a recent study into indirect genetic effects. Now, traditionally, if you're interested in understanding how much of the differences in individuals in a population, a particular trait, are due to genetic differences, you're looking for direct genetic effects. And the concept of indirect genetic effects takes us a little bit further. So if you look at this graph here over on the left, you see we've got a particular trait here, and it's of course affected by a genotype G and its own environment. But you see this component down here shows that part of the environment in social animals is given by social interactions. And so therefore, we have this indirect genetic effect component. So in other words, we're interested in how do genes expressed in social interactions, such as siblings, such as mothers, etc., influence trait expression in a focal individual. And that's important, of course, not just to understand exactly where the sources of variation in the trait come from, but of course it has also very important evolutionary implication. It's been well established for two, 3 decades now. Jason Wolf, Alan Moore and colleagues have shown towards the end of the 90s and 2000s that if you include indirect genetic effects, and of course this graph is showing you for the most important indirect genetic effect, namely maternal genetic effect, then the evolutionary implications can be very profound. So the most important indirect genetic effect, as I said, are maternal effects, of course, because here we're looking at genes expressed in mothers. They influence somehow how mothers behave towards their offspring, and that in turn influences offspring traits. If we're looking at this graph here, what happens in evolution in terms of if we are looking at the case of the blue line here, we have the absence of indirect effects. We've got a period of selection here of 5 generations. We see this is the predicted relationship between genotype and phenotype. But if we're having positive or negative indirect genetic effects here in this case, we see there are profound differences in the genotype-phenotype relationship. So that's why indirect genetic effects are very, very important for social organisms, and in fact most of the organisms, of course, are social. And so specifically the question we were interested in answering is, can we find genes that are expressed in offspring that influence the quality of maternal behaviour, and if so, what is the mechanism by which that may happen? And so the model system that we are using is the largest mammalian genetic reference system. It's the BXD mouse population. It's a recombinant inbred mouse population that basically was generated by crossing 2 inbred strains of mice. On the top left corner here, the black mice and the red mice, grey mice. It's a cross between black 6 and DBA, and of course these are inbred mice. There's no genetic variation within each of these populations. They're being intercrossed, and then you get these groups of F2 animals here, and then you interbreed within each of these groups to generate your lines down here. And so basically what you do is, in essence, you're freezing, and I could actually do this over here much better. You're freezing a particular recombination pattern in these lines. So these differently colored lines represent different genotypes within the BXD population. So within each of these lines, there's no genetic variation within amongst all the animals, but of course across the BXD population, if you're looking here at this example, you see, for example, here you've got a red allele, a black 6 allele. You go down the same genomic location, you've got a D2 allele, and so on and so forth. So that's the genetic variation that we're exploiting in our model system, in our experiment. And here's some examples of the BXD mice. You see they're not just very diverse, of course, in coat color, but they're highly diverse in all sorts of behavioral traits and in developmental traits, although they haven't actually been selected for any particular trait. And the range of the traits is also much greater than the original 2 founding strains. And then the method that we're using is simply interval mapping. I don't think I need to explain that to the audience here. We're basically looking for an association between genetic variation and phenotypic variation in our population. But the key point that I want to make is the experimental design, and that's the difference to, of course, a traditional mapping experiment. What we're setting up is an experimental population of adoptive mouse families here. So here you've got, in black, our black 6 mice. Remember that these are inbred strains and there's no genetic variation amongst them. And here, the different shades of grey represent the different BXC genotypes. And so what we're doing is we're breeding black 6 males and black 6 females, we're getting the litters, we take them away, and we're giving these mothers then different litters of different BXC genotypes here. And if you're looking at this scenario here, we see that in this population, all of the mothers have the exact same genotype. There's no genetic variation amongst them because they're all genetically uniform. But they're nursing offspring of different genotypes here. And so if we're interested in understanding variation, for example, in maternal behaviour, or measuring maternal behaviour in these mothers here, we know that these differences, that any differences cannot be due to differences in their genotype, because there aren't any differences between their genotype. And instead what we predict is any differences in maternal behaviour in these mothers here are due to the differences in genotype in their adoptive offspring that makes them behave somewhat differently towards their mothers, and mothers respond differently. So, in other words, we are mapping variation in maternal traits as a function of offspring genotype, and of course, controlling for the effects of maternal genotype and reverse. Of course, we can do over here as well. And so the experiment is fairly straightforward. We're setting up these cross-fostered families, we're measuring behavior and development during the first 3 weeks of lactation. We're then calculating the strain averages for each of the genotypes, or for all of the traits that we're interested in. And of course, we also need to statistically adjust for confounding factors. If you remember the previous example with all the mothers, I said there are no genetic differences, of course, but they can be different, of course, due to the differences in body weight, and therefore they may behave differently, so we need to statistically adjust for that, and then we're mapping basically the variation, the residuals in this case, in these traits as a function of their offspring's genotype, and not as a function of their own genotype, as what you would do in a normal analysis. And so this is exactly what we're doing here. Here we're looking at mapping variation in maternal nest building behavior on day 6 of the phenotype, and on 3 days during lactation. We scored nest building behavior in these mothers here, and then we're mapping variation in nest building behavior as a function of their own of their adoptive offspring. And what we find here is a locus on the chromosome 7, where we basically have within this region here on chromosome 7 one or several candidate genes where allelic variation contribute to variation in maternal behavior, not necessarily offspring behavior. So we don't know exactly what the direct effect is of this locus, but it is very clear that maternal behavior is affected by genetic variation in their offspring genotype. And of course, we can do the reverse. This is simply mapping maternal genetic effects. Here we're asking the question, how much variation in this case in offspring growth is due to genetic variation in their mothers, and that's a sort of the next surprising result. So here we find a locus in chromosome 7 and possibly in chromosome 2 as well, where allelic variation basically influences how well their offspring are doing. Okay, so now that we've established that variation in maternal behavior is affected by genetic variation, we can look at, well, how does it actually work? And so then we're looking at solicitation behavior, that is how offspring demand resources and how responsive their mothers are. And we find in these adoptive families here, we find there's a positive correlation between solicitation and maternal behavior. So the more basically offspring demand, the more their adoptive mothers are willing to give, so to speak. Okay, so that's interesting for starters. But possibly more interesting is to look at the correlation within biological families. Remember that we were cross-fostering the animals, so we can ask basically what's the correlation between maternal behavior of a particular BXD genotype and the offspring behavior of her biological litter, of course, which gets nursed by a black 6 mother. And when we're looking at this correlation, remember that in all of these scenarios we're controlling for genetic variation in the other partners, so either in mothers or in offspring. And so here what we are finding is if we are correlating, in this case here, short-term weight gain provisioning basically, we find actually a negative correlation between solicitation and maternal behavior. So in other words, mothers that are particularly generous produce offspring that are weak elicitors. And that we have labeled as an indication for co-adaptation, because of course it's not a random association. Okay, so we find that in our adopted families, the more offspring solicit, the more they get, but does it actually pay off? And so we're looking then at the correlation between solicitation and growth. And actually, as it turns out, the more they ask for food, the less they grow. And we believe that this is due to the increased cost of solicitation, basically what it entails in mice that are running after their mothers, basically are very demanding and possibly expending quite a lot of energy just showing this particular behaviour. Okay, so what we've seen is that the mothers respond to offspring solicitation, these adoptive families. That was the first result, and if we're looking at the biological families, we find there's a negative correlation between growth and solicitation. However, although increased solicitation results in increase in maternal behaviour, which is all good, it does not pay off for offspring. Excuse me. They have less growth, and that may well be due to the cost of solicitation in energetic terms. Okay, so just to summarise very generally our results, we find clear evidence for indirect genetic effects in offspring. That is, we find evidence for genes that are expressed in offspring that modify maternal behaviour, and therefore, of course, indirectly also offspring fitness. We find that maternal provisioning and offspring solicitation show signs of coadaptation in their biological families. The main problem with the study, caveat here, is that we cannot control for intrauterine maternal effects. So remember that the way that we set it up is, of course, we want to control for genetic variation either in the parent or in the offspring. We can do this all once the pups are born, but we can't do very much about intrauterine maternal effects. It's just experimentally not feasible in a genetics experiment. And next, of course, we would like to identify the pathways from these direct genetic effects to indirect genetic effects. So that's the key missing thing here. Do you remember the first genome scan I showed you that influences maternal behaviour? Of course, it has to do something to the offspring behaviour that then has a good effect onto the maternal behaviour. So that's something that we now need to look at in a mixed litter design, and hopefully I can present some results of that at the next meeting. So with this, I'd like to conclude and just thank my collaborators and the various funding agencies. And I believe we've got 2 minutes, 1 minute for questions. So if you've got any questions, please, yeah,